Good morning, students.
Good morning, ma'am.
Yes, still you people are joining. Only 50. Why? What happened to you? Out of 150, only 50 has joined till now. Hmm? That to 10 o'clock class. Yes, any problem? Hmm? Any problem with you people? No response, that is the biggest problem. Hmm? Yes. So, what are we discussed in the last class? Anyone? What all we discussed in the last class? Yes? Anyone from out of 60 students? What we discussed in the last class? Which system we discussed? Hmm?
Respiratory system.
Yeah, respiratory system. So in that, what are the subtopics we discussed? Hmm?
Compliance.
Yeah, complaints of the lungs. Last class I have not discussed complaints. Hmm? Last class, what was the thing? Maybe revision of compliance. Yes. Then V by P ratio, isn't it?
Dead space.
Dead space. Yes, very good. Then diffusion capacity of lungs. Yes, Srushti, you want to say something? Yes. So today in this class we are going to discuss about the next competency, that is 6.3 competency in the transport of oxygen and carbon dioxide I am going to cover. Okay. So you know already the primary function of our lungs is to take oxygen from the environment, okay, and return back, expel carbon dioxide which is producing continuously in our body because of the metabolism to outside, okay, removing carbon dioxide and supplying oxygen. So in lungs, the oxygen is taken from alveoli to the pulmonary capillaries, okay, and carbon dioxide is diffused from pulmonary capillaries to alveoli, isn't it? After this first stage of gaseous exchange, these oxygen has to be carried to each and every cell of a body, and carbon dioxide has to be carried from the cells to the lungs, isn't it? So this is also one of the very important component of our respiratory system, that is transport of oxygen and carbon dioxide in the blood. Okay, so this is done mainly by the blood. Okay, some amount in the plasma and more amount in the hemoglobin. So this is what you are going to learn in today's class, how oxygen is carried from lungs to the each and every cell of our tissues, and how the carbon dioxide is carried from each cells and tissues to the lungs. Okay, that's what we are learning in today's class. So you already know the mechanics of breathing. So what all things will happen during inhalation and what all things will happen during exhalation. These are my very preliminary and initial classes, isn't it? So inhalation, diaphragm will contract, isn't it? Diaphragm will contract and inspiratory muscles will contract and rib cage will expand and the air enters into the lungs. Okay. Whereas in exhalation, diaphragm relaxes. Inhalation is an active process, whereas exhalation is an passive process. Isn't it? So active active contraction of the muscle occurs in inhalation, and relaxation of same muscle will lead to the exhalation at rest. Okay, at rest we did not need any energy for exhalation. Hence, exhalation is a passive process. These things in detail I have told you already. The bucket handle movements, pump handle movements. Okay, these all things comes under mechanics of breathing. Okay. Now, coming on to the distribution of oxygen in the body. Okay, so what is the partial pressure of oxygen in different parts? Like you might be knowing already these things. Okay, in inspired air, partial pressure of oxygen is 158 mm of Hg. Okay, and oxygen content of the inspired air for 100 ml it is 21 ml of oxygen is present. Okay, DL is nothing but deciliter or 100 ml. Okay, for expired air, partial pressure of oxygen is 116 mm of Hg, whereas the oxygen content is 16 ml per DL. Okay, for 100 ml of expired air, 16 ml oxygen is present. Okay, then alveolar air, the partial pressure of oxygen is 100 to 104. Okay, whereas the oxygen content is 13 to 14 ml per dl. And in arterial blood, it is around 98 to 100 mm of Hg, whereas in oxygen content is around 19 ml per dl, and in venous blood the partial pressure of oxygen is 40 mm of Hg, whereas the oxygen content per ml of 100 ml of venous blood is around 14 ml. Okay, so these are the partial pressure of oxygen and oxygen content in different parts like inspired air, expired air, alveolar air, arterial air and venous blood. So alveolar air PO2 is 100 to 104. So actually logically if we think arterial blood PO2 also should be same like that of the alveolar air, but it is less. It is because of the admixture of some venous blood, like from some Thebesian vein, cardiac vein, directly poured into the pulmonary capillaries. Okay, so there is an admixture of some amount of venous blood, hence arterial blood PO2 is not exactly that of the alveolar air. Okay, so with this, we can come to know that for each 100 ml of inspired air, 5 ml, that is 21 minus 16 ml of oxygen is extracted by the blood, isn't it? For each 100 ml of inspired air, 5 ml of oxygen is extracted by the blood, and for each 100 ml of arterial blood, 5 ml of oxygen is extracted by the tissue. Is it right? For each 100 ml of inspired air, 5 ml of oxygen is extracted by the blood, and for each 100 ml of arterial blood, 5 ml of oxygen is extracted by the tissues. Okay. Now coming on to with that introduction, coming on to the transport of oxygen. Okay. So, oxygen is transported in two forms. First is physically dissolved form in plasma, and another one is chemically combined to hemoglobin. So, in physically dissolved at normal body temperature, 1 ml of plasma contains 0.00003 ml of oxygen per mm of Hg PO2. Okay, that means normal arterial blood contains 0.3 ml of oxygen per 100 ml. Okay, so normal arterial blood contains 0.3 ml of oxygen in a dissolved form. Okay, per 100 ml. In case of combined with the hemoglobin, 1 gram of hemoglobin can combine with 1.34 ml of oxygen when fully saturated. Okay, 1 gram of hemoglobin can combine with 1.34 ml of oxygen when fully saturated. So a person normally with 15 gram of hemoglobin per 100 ml of blood can carry 20 ml of oxygen per 100 ml of blood. Okay, so if 1 gram it is 1.34 ml, then for 15 gram if you multiply 15 into 1.34, you will get around 20 ml of oxygen per 100 ml of blood. Okay, so, as I told you already, oxygen is carried in blood in two forms. That is, 97% maximum amount of oxygen is carried bound to hemoglobin, whereas 3%, okay, only 3% of oxygen is carried dissolved in the water of plasma and blood cells. So, only 3% of oxygen is transported in a dissolved form in plasma. Okay, and hemoglobin increases the oxygen carrying capacity of blood to 30 to 100% because of the presence of hemoglobin in the blood, okay, or in the RBC. Oxygen carrying capacity has enormously increased. The transport of oxygen and carbon dioxide by the blood depends on diffusion and the flow of blood. Okay, so oxygen and carbon dioxide transport depends on the rate of diffusion and the intactness and normal blood flow to that particular tissue. Okay, so now coming on to the role of hemoglobin in oxygen transport. As I already told you, 97% of oxygen is carried in chemicals combined with hemoglobin in the RBC, and 3% is transported in dissolved state in water of plasma and blood cell. Thus, under normal condition, oxygen is carried to the tissue almost entirely by hemoglobin. Okay, so the oxygen molecules combines loosely and reversibly with the heme portion of hemoglobin. So here one more point you must know. You must recall the hemoglobin structure. You know the hemoglobin structure, how it is? What's the structure of hemoglobin and how many iron atoms are present in one molecule of hemoglobin? Anyone? Hmm? How many ferrous ions are present in one molecule of hemoglobin? Yes?
One.
Yes? Anyone?
One.
One? No? No? How many iron molecules are present in one? Yes, four. Very good. Four. How I will tell you. Okay. So when PO2 is high, as in the pulmonary capillaries, that is partial pressure of oxygen is high in the pulmonary capillaries, oxygen binds with the hemoglobin. When partial pressure of oxygen is low, as in the tissue capillaries, oxygen is released from the hemoglobin. Okay. So whenever partial pressure of oxygen is more, as in the lungs, oxygen binds to the hemoglobin. Whenever partial pressure of oxygen is less, as in the tissue capillaries, oxygen is released from the hemoglobin. Okay. So this is the structure of your hemoglobin. So it has two alpha chain and two beta chains, isn't it? And it has the four iron atoms. In one molecule of hemoglobin has four iron atoms. Okay, and one atom of iron combines with one molecule of oxygen, that is O2. Okay, O is considered as two atoms of oxygen or one molecule of oxygen. You must be clear here. If you are talking about the molecules, then one iron combines with one molecule of oxygen. If you are talking about the atoms, one molecule of iron combines with two atoms of oxygen. Okay, so the one molecule of hemoglobin now carries 4 O2. Is it correct? 4 molecules of oxygen. One molecule of hemoglobin carries 4 molecules of oxygen, or one molecule of hemoglobin carries 8 atoms of oxygen. Okay, I hope there should not be any confusion Here, you must remember, go back to the hemoglobin structure, read once again what is the structure of hemoglobin and which form iron is present. Okay, that's not my topic today. So, what I want to tell you here is hemo iron. So, one iron combines with one molecule of oxygen. So, once one iron atom combines with one molecule of oxygen, the affinity of another molecule for oxygen increases. Okay. So, for third molecule, it is further increased. For fourth molecule, the affinity of hemoglobin to the oxygen goes on increasing from the first iron atom to the last. Okay, and the oxygen is combined with the iron. It is called as oxygenation, not oxidation. Okay, so oxygenation means it is loosely, temporarily and reversibly attached so that it can be easily released at the level of the tissues. Whenever we require oxygen, it should be easily released from the oxygen. Hence it is called as oxygenation, not oxidation. When we say oxidation, when ferrous iron is converted to ferric, then we say the oxidation. But here oxidation is not occurring, only the ferrous iron atom only is combining with the oxygen molecule with loosely and reversibly. Okay, so this must be understood very clearly. Okay, now next coming on to the very important part of oxygen transport, that is oxygen hemoglobin dissociation curve. It's very important and it's very frequently asked. Almost all question paper you search, this question is there. Okay, so draw the oxyhemoglobin dissociation curve and write the details. So here what we have taken, partial pressure of oxygen in the x-axis and hemoglobin saturation in percentage, that is percentage of hemoglobin saturation in the y-axis. So if we draw a plot, we will get a graph of sigmoid shape. It is not linear, isn't it? It is not totally linear, but we are getting the sigmoid type of curve. Why? What is the reason for sigmoid shape of oxyhemoglobin curve? Is, as I told you, the affinity of oxygen goes on increasing when oxygen started combining with the hemoglobin. Starting, if it combines with only one atom of iron, its affinity is less. But once it is attached to one, the second molecule has more affinity. Very fast it will attach to the another molecule. For third molecule, it will attach very fast for the third molecule. So the affinity of oxygen, hemoglobin for oxygen goes on increasing as the one by one oxygen molecules are attaching. That is the reason we will get the sigmoid curve. So here we have the two part of curve. Okay. So first part is we will get the steep part. Okay. So this is very less change in the partial pressure of oxygen, there is an enormous change in the oxygen saturation. This is called as a steep phase of a oxyhemoglobin curve. But if you come to this level almost, okay, suppose from 80, okay, 80 or 70, if you take, or around 60 only, okay, if you take 60 to 100 also, there is not much change here. It is almost like a plateau stage, isn't it? So from 60 to 100, there is not much change in oxygen saturation. So if you consider at 60 also, see how much is oxygen saturation? 90%. 90% hemoglobin saturation occurs. Okay, at PO2 60 also. That means hemoglobin is 90% combined with the oxygen here. That means if the PO2 level has decreased to 60 also, 60 nm of Hg also, our hemoglobin will be 90% saturated. This gives an important, this is one of the very important factors. So PO2 maybe go on decreasing if you go to somewhat high altitude or if you go to the deep sea. Okay, so the PO pressure of PO2 may be changing. So if there is an enormous change from 60 to 100 mm of Hg also, there will not be much change in our oxygen saturation, and this prevents our cells in hypoxia, isn't it? So if our PO2 would have changed to 80, okay, then suddenly if this mechanism would not have been there, then our tissue or cells might have suffered from the hypoxia. Okay, this is a protective phenomenon. Okay, this is the oxygen reserve which is present in our blood, okay, in our hemoglobin, so that around 60 to 100 mm of Hg, the curve is in a plateau stage. There is not steep change here. Okay. So that means the oxygen hemoglobin saturation remains 90% even if the PO2 is 60%. That is the physiological significance of this plateau phase. You must understand this. Then what is this steep phase indicates? This steep phase indicates, so in a tissue level, okay, the PO2 will be very less, almost around 40, 50, isn't it? So PO2 will be around 40. So here the still little change from 40, enormous amount of oxygen will be released into the tissue because the curve is very steep here, isn't it? That's what indicate. This happens at our tissue levels. So very less change. Okay, if it comes to the 40 to 39 also, then it meaning that tissue is going for hypoxia and enormous amount of oxygen is released from our tissue. Okay, so this happens at our tissue levels, and this phase usually occurs at our lung level. Okay, so here the oxygen combines with the hemoglobin here. Okay, so this is about the oxyhemoglobin dissociation curve. Okay, the curve is sigmoid shape. Okay, because of differing affinity of hemoglobin for oxygen, it is of sigmoid shape. It has two parts, steep part and the plateau part. Okay, the physiological importance of plateau part is if the PO2 ranges from 60 to 100 also, enormous increase in the change in the PO2, but our hemoglobin remains, so hemoglobin saturation remains up to 90%. That is the importance of our plateau phase. And very important, one more point here is there is a P50 value also. Okay, so what is P50 value? So when PO2 is 50 mm of Hg, how much the oxygen is saturated? That is called as P50 value. So if this curve is shift to the right, okay, P50 value will increase. Yes or no? If this curve is shift to the left, the P50 value will decrease. Okay, isn't it? Okay, that's what the meaning. So that is also one of the very important factor you must know. What is the P50 value? So if the curve shifts to the left, P50 value will decrease. Okay, not increase. Okay, so now next coming on to the factors that shift the oxygen hemoglobin dissociation curve. Okay, this is also very important factors. So as I told you, the shift may occur to the right or the shift may occur to the left, isn't it? So yes. So shift to the right occurs in these conditions. That is increase hydrogen ion or decrease in pH. So as it has been shown, if pH is decreased, it has shifted to the right. If pH is increased, it has shifted to the left. Okay. So shift to the right occurs whenever there is an increased hydrogen ion or less pH, decrease in pH, and increase carbon dioxide. Increase carbon dioxide again lead to decrease in pH, isn't it? And increase temperature. Increase temperature also causes shift of the oxyhemoglobin curve to the right and increase 2,3 dpg. It is given in your books. Okay, biphospho-glycerate it has given here, but in your books it has given 2,3 dpg. Both are same. Okay, 2,3 diphosphoglyceride. Okay, both are same. No problem. Okay. So increase to 3 dpg. So shift of right occurs in these conditions. So how to remember these? You can imagine during exercise. Okay, exercise, temperature of the body increases, increased carbon dioxide production occurs because of the increased metabolism, increased 2,3 dpg occurs and pH will be reduced. So you remember in that way. So during exercise, what all changes will occur that cause the shift of oxyhemoglobin curve to the right. Okay. So what is it importance here is when the curve is shift to the right, the affinity for hemoglobin for oxygen decreases. That means the oxygen can be easily released to the tissues. Okay, oxygen from the hemoglobin can be easily released to the tissue. So this occurs in the tissue levels or active levels or exercising levels. Okay, and in reverse, suppose the pH is increased or decrease in hydrogen concentration, decrease in CO2, okay, decrease temperature and decrease to 3 dpg. So reverse of these condition causes shift of the curve to the right. Okay, shift of the curve to the right. And what is the meaning of shift of curve to the right? It causes the affinity for hemoglobin for oxygen increases. Okay, so this usually occurs in the lung level because oxygen has to combine with the hemoglobin, isn't it? So whenever there is a PO2 will be more, the affinity for oxygen to bind hemoglobin, affinity of hemoglobin to bind with the oxygen increases. It is not easily released. That's what happens if reverse of this condition will occur. Okay, so this occur at our lung level and this occurs at our right shift occurs at our tissue level, left shift occurs at the lung. You are understanding this? Any problem, students? You are able to
No,
understand?
ma'am.
Huh?
Yes, ma'am.
Okay. So these are the very important factors you must remember. What are the causes of right shift of the oxyhemoglobin curve and what are the causes for the left shift of the oxyhemoglobin curve and what is its physiological importance. You must understand and remember this. Okay, when the curve is shifted to the right means affinity for hemoglobin for oxygen decreases and oxygen is readily released to the tissue. When the shift of left occur means affinity of hemoglobin for oxygen increases. Okay, it is easily cannot be released from the hemoglobin but it facilitate binding of oxygen with hemoglobin. Okay, so these are the causes for right and left shift of oxyhemoglobin curve. Next, coming on to the Bohr effect. What is this Bohr effect? A shift of the curve to the right in response to increase in blood carbon dioxide and hydrogen ion. So whenever there is an increase in the CO2 and increase in the H plus ion, the curve will shift to the right. This is called as Bohr effect. Okay. So has a significant effect, enhances the release of oxygen from the blood in the tissue. So it causes the release of oxygen from the blood and enhances the oxygen of blood in the lungs. Okay. So this causes oxygenation increases in the lungs. Okay. And it facilitates release of oxygen at tissue level. So this is called as Bohr effect. So increase in carbon dioxide in the blood and increase in H plus ion causes release of oxygen from the blood in the tissues, and it also helps in oxygenation of blood in the lungs. Okay. In the tissue, carbon dioxide diffuses from the tissue cells into the blood. This you know. Because of the metabolism, carbon dioxide will be producing. It diffuses from the tissue into the cell. This increases the blood PCO2, which in turn raises the blood carbonic acid and the hydrogen ion concentration. These effects shift the curve to the right and downwards. Okay. In the lung, what happens? The carbon dioxide diffuses from the blood into the alveoli. So the carbon dioxide carried to the lungs for expulsion, isn't it? Diffuses from the blood into the alveoli. This reduces the blood PCO2 and decrease the hydrogen ion concentration, shifting the curve to the left and upward. That means in the lung it facilitates oxygenation of hemoglobin, binding of oxygen with the hemoglobin, whereas at the tissue level it facilitates release of oxygen to the tissue. Okay, so this is called as Gouhar effect. Next, coming on to the diffusion of oxygen from alveoli to the pulmonary capillaries. So what happens? So pulmonary arterial end. So arterial end, the PO2 is 40 mm of Hg. Okay. So here but alveolar PO2 is already 104 mm of Hg. So because of this gradient, oxygen enters into the pulmonary capillaries. Okay. And the almost it is equal to the alveolar PO2, that is 104 mm of Hg at the venous end, but it further decreases because of the admixture of some venous blood directly, that is cardiac vein, some of the Sebacean vein directly pours, and hence our arterial PO2 is around 98 to 100. It is not 104. That's what I told you, isn't it? So in case of pulmonary capillary, the blood enters, that is arterial end, PO2 is be 40, and in the venous end, PO2 will be 104 mm of Hg. Okay, so the diffusion occurs at a initial pressure difference that causes oxygen to diffuse. That causes oxygen diffuse into pulmonary capillaries 104 minus 40 are 64 mm of Hg. So this much pressure difference is there in our pulmonary capillaries for diffusion of oxygen. Okay. Next, coming on to the diffusion of oxygen from the pulmonary capillaries into the tissue fluids. So in the tissue, okay, so PO2 is around 23 mm of Hg. Okay. In case of arterial end of the capillary, the PO2 is 95 mm of Hg, as you know already these things. Okay. So here what happens? Here the cells will take. So arterial capillary has a PO2 of 95 mm of Hg. Okay, so the cell has a PO2 of 40. So the diffusion of oxygen from the blood to the cell occurs here. Okay, cell occurs, and finally the arterial pH PO2 becomes 23 mm of Hg. Okay, and venous end of capillary blood, it becomes, after giving the oxygen, it becomes around 40 mm of Hg. So diffusion of oxygen from the peripheral tissue capillaries to the cell. So PO2 in interstitial fluid is 40 mm of Hg. This what? Okay, 40 mm of Hg, and tissue it is 23 mm of Hg. So from arterial capillaries to interstitial fluid, diffusion occurs. From interstitial fluid to cells, the diffusion occurs. Finally, the arterial PO2 95 mm will become venous end PO2 of 40 mm of Hg. Okay, this is the diffusion of oxygen from peripheral capillaries into the tissue fluids. Okay, so this is change in PO2 in the pulmonary capillary blood, systemic arterial blood and systemic capillary blood, demonstrating the effect of venous admixture. As I already told you this, so systemic venous blood, as I have shown you, okay, it is 40, PO2 is 40. So in the lungs, it attains as that of the alveolar, okay, that is 104. But because of the admixture of some of the venous blood directly, the systemic arterial blood is around 98 to 100. PO2 is 98 to 100. Then it goes on decreasing in the systemic capillaries and systemic veins. It come back to the 40. Okay. So this what will happen, changes in PO2 in the pulmonary capillary blood, systemic arterial blood and systemic capillary blood. Okay. Now coming on to the transport of oxygen in the dissolved state. Okay, as I told you, the amount of oxygen transport in the dissolved state is only about 2 to 3% of the total. But during exercise, when hemoglobin release of oxygen to the tissue increases another threefold. So this is the what is the importance of dissolved oxygen is, it's even though it's transported in a very minute amount, but during exercise this is the pathway where the oxygen can be dissolved in a more amount and can be increased. But oxygen, in case of oxygen, it cannot be increased. Why? Means oxygen has a, if it has saturated to 100%, how much ever you increase your PO2, you cannot increase the oxygen level, isn't it? But in the dissolved form, when you increase in the PO2, corresponding to the increase in the PO2, increase in the dissolved form of oxygen occurs. Okay, that you must understand very clearly. So during exercise, what happens? There is a threefold increase in the dissolved oxygen. Threefold increase in the transport of oxygen can occur in a dissolved form, and this oxygen transport in the dissolved state falls to as little as 1.5%. That means more amount of oxygen is extracted and this dissolved form may decrease. Okay. Then oxygen poisoning, you must know this also. A condition happens if a person breathes oxygen at very high alveolar PO2 level, so the amount transported in the dissolved state can become much greater, so that a serious excess of oxygen occur in the tissue. Okay, that is called as oxygen toxicity or oxygen poisoning. Okay, now coming on to the combination of hemoglobin with carbon monoxide. Okay, why it is important? Because carbon monoxide combines with hemoglobin at the same point on the hemoglobin molecule as does oxygen. So it can displace oxygen from the hemoglobin, thereby decreasing the oxygen carrying capacity of blood. But it binds with 250 times as much tenacity as oxygen. So its affinity for oxygen, okay, hemoglobin affinity for carbon monoxide is 200 times more compared to the oxygen. So if oxygen and carbon monoxide are available in the same amount, the carbon monoxide has 200 times more affinity for hemoglobin, so it will go fast and it will bind to the same side where oxygen has to bind. Okay, so the hypoxia may occur. So the curve is almost identical to the oxyhemoglobin dissociation curve, except that the carbon monoxide partial pressure are at a level of 1 by 2/5 of those for the oxygen hemoglobin dissociation curve. You can make out here. The pressure is in points. Okay, 0.1, 0.2, 0.3 and 0.4. So at 0.4 mm of Hg of PCO, partial pressure of carbon monoxide, the hemoglobin is 100% saturated with carbon monoxide. Okay, you can make out. So you must remember this point in carbon monoxide poisoning. It's very important in clinical practice. Carbon monoxide is nothing but it is present in your smoke. Okay, so whenever something is burning, okay, the smoke will have a maximum amount of carbon monoxide. Okay, so gas, you must remember this. So carbon monoxide has 250 times more affinity to bind with the hemoglobin in the same site where oxygen can be bind. So how you treat this patient? Okay, whenever there is a carbon monoxide poisoning, immediately you have to give oxygen, 100% oxygen. Okay, so if the oxygen concentration is very high, it may displace the carbon monoxide from the same site. 100% oxygen is the treatment for carbon monoxide poisoning. Okay, so and they said when PCO2 increases to up to 0.6, that may be lethal. Okay, death of the patient may occur if the PCO, partial pressure of carbon monoxide in our blood, increases up to zero point 0.6 mm of Hg. Okay, so that's what is written here. Carbon monoxide partial pressure of only 0.4 mm in the alveoli allows the carbon monoxide to complete equally with the oxygen for combination with the hemoglobin. Therefore, the pressure of only 0.6 mm of Hg can be lethal. Treatment is giving 100% oxygen. Now, so this is the summary of what I told till now. Okay, carriage of oxygen in the blood. So in arterial blood, its PO2 is around 100 mm of Hg. Oxygen content is around 90 ml per dl, 100 ml of blood. Form in which oxygen is carried. So 0.3 ml per dl is carried in a dissolved form, that is dissolved in a plasma, and out of this 19 ml, okay, dissolve 18.7 ml per dl in bound to hemoglobin, that is 97% is bound to hemoglobin and 2% is in a dissolved state. That's what I told you. So around 19 ml, 0.3 ml is carried in a dissolved form and 18.7 ml is carried in a combined with hemoglobin. In case of a venous blood, PO2 is 40 mm of Hg and oxygen content is around 14 ml. So here 0.13 ml per 100 ml is in a dissolved form and 13.8 ml per 100 ml is transported in a bound to the hemoglobin. Okay, so now amount of oxygen held by different vehicles. This also the revision of previous slides what I told you. So in plasma at PO2 100 mm of Hg, how much it carries? As I told you, 0.3 ml per deciliter. Okay, in venous blood, that is PO2 at 40 mm of Hg, so it carries around 0.12 ml per dl. This I told you in the previous slide. So given to the tissue is how much? Around, combining these two, how much it is? 0.18 ml per dl. So this much is not sufficient. Okay, so in a plasma or in dissolved form, in arterial blood it will be this much, in venous blood it will be this much. Okay, so it given to the tissue. Okay, this minus this. Okay, zero point 0.3 minus this, it is not sufficient. Combining with hemoglobin at arterial blood, 19 to 19.5 ml per dl is oxygen is carried combining to the hemoglobin, and venous blood it is around 18 ml per dl. So it is given to the tissue is around 1 to 1.5 ml per dl. This is also not sufficient. So in blood total, okay, in total blood, so in arterial blood the oxygen carries 90 ml, 19 ml per 100 ml, and in the venous blood it is 14 ml per dl. So difference is 5 ml per dl. So blood is the ideal vehicle for transport of oxygen, not only the plasma, not only the hemoglobin, both of them together, that is in a blood, is a ideal vehicle for transport of oxygen. Okay. So now these are some of the points to be remembered. That is, each hemoglobin can carry 4 oxygen molecules. Okay, and one RBC contains around 250 million hemoglobin molecules, so can carry 1 billion of oxygen. Okay, 1 gram of hemoglobin combines with this also I told you 1.34 ml of oxygen. Hence, the 100 ml of blood contains 15 gram of hemoglobin, so it will carry 20 ml of oxygen. So these points you are supposed to remember. Okay. Now there is one condition called cyanosis, which occur when more than 5 gram of hemoglobin per 100 ml of arterial blood are in a deoxy state. Okay. So when oxygen is combined to the hemoglobin, it is called as oxyhemoglobin. When the hemoglobin is removed from the oxygen is removed from the hemoglobin, then it is called as deoxyhemoglobin. Okay, that are reduced hemoglobin. So if there is a reduced hemoglobin is more than 5 gram per 100 ml of arterial blood, then a bluish discoloration of body and mucous membrane occurs. That condition is called as cyanosis. Absence of cyanosis does not rule out hypoxemia, and presence of cyanosis. So cyanosis will not occur in anemia because hemoglobin level itself will be less. Okay, so that does not rule out hypoxemia, and presence of cyanosis does not always indicate hypoxemia. Okay, so this you can read cyanosis in detail. Okay. Next, coming on to the transport of carbon dioxide in the blood. So again, the carbon dioxide is transported in a three ways. That is physically dissolved form, okay, carbamino compounds, and as a bicarbonate. So carbon dioxide is transported mainly in a bicarbonate form. That is 80 to 90% is transported in a bicarbonate form. So at 37, that is body temperature, 1 ml of plasma contains 0.0006 ml of carbon dioxide per mmHg PCO2. Normal arterial blood contains 2.4 ml of carbon dioxide per 100 ml. Okay. Now, transport of carbon dioxide in the blood. So, arterial and PCO2 is around 40 mm of Hg. Okay, in interstitial fluid it is 45 mm of Hg, where inside the cell it is 46 mm of Hg. Okay, so there is a pressure difference of only 5 to 6 mm of Hg, whereas the oxygen is, for oxygen transport the pressure difference is around 64, isn't it? 64 to 65 mm of Hg, but for carbon dioxide only 5 to 6 mm of Hg present. Pressure difference is enough for transport of carbon dioxide from the tissues to the blood. Okay, that means the diffusion capacity of carbon dioxide is more compared to the oxygen. So they say that the diffusion capacity of carbon dioxide is 20 times more than the oxygen. Okay, so uptake of carbon dioxide by the blood in the tissue capillaries, PCO2 in the tissue is 46, interstitial fluid is 40. So the arterial end of capillary PCO2 is 40. After passing through the cell, it coming in the venous end of capillary, it increases to 45 mm of Hg. Okay. Now, transport of carbon dioxide in the blood. Okay. So, same thing. Okay, arterial blood it is 45 mm of Hg, whereas in the pulmonary capillaries, okay, pulmonary capillaries it is around, it is actually transport of carbon dioxide in the lungs. Okay, So PCO2 in the alveolus is around 40 mm of Hg. Hence, the PCO2 readily diffuses to the alveoli. Okay, and it is exhaled from the lungs. Okay. So PCO2 of the blood entering the pulmonary capillary at the arterial end is 45 mm of Hg and PCO2 of the alveolar end is 40. So only a 5 mm of Hg pressure difference causes all the required carbon dioxide diffusion out of the pulmonary capillaries into the alveoli. Okay. So now coming on to the carbon dioxide diffusion occurs in the direction opposite to the diffusion of oxygen. Okay. So carbon dioxide can diffuse about 20 times as rapidly as oxygen. Therefore, the pressure difference required to cause carbon dioxide diffusion are less than the pressure difference required to cause oxygen diffusion. Okay, you understood? Only a pressure difference of 5 mm of Hg is enough for the diffusion of carbon dioxide compared to the oxygen. Okay. Now coming on to the what are the different ways carbon dioxide is transported. So as I told you, dissolved around 7%, bound to the hemoglobin, that is carbaminohemoglobin is around 23%, and as a bicarbonate form around 70%. And some book they will say it is 80% to 90% is transported with as a bicarbonate form. Okay. Then transport of carbon dioxide in the form of bicarbonate ion. So this is the major way of transport of carbon dioxide, that is as a bicarbonate ion. Okay. So carbon dioxide is generated in the tissue and diffuses freely into the venous plasma and then into the RBC. Okay. So it is produced because of the metabolism and venous plasma and then into the RBC. In the RBC, carbon dioxide combines with water to form carbonic acid. In RBC, carbonic acid is in a carbonic anhydrase enzymes are present in abundance. So in the presence of those enzymes, carbon dioxide combines with water to form carbonic acid, a reaction that is catalyzed by carbonic anhydrase. These enzymes are maximum in the RBCs. Then the bicarbonate ion dissociates into H plus and CO3. So this is a parent reaction or major reaction. You must remember this occurs in our RBCs. Okay. Now coming on to the transport of carbon dioxide in the blood. So what happens here? From the cells, carbon dioxide is produced because of the metabolism. It enters into the blood and into the RBCs. Okay. Some amount of carbon dioxide is transported dissolved in the plasma, isn't it? That is around 7%. Okay. Some amount combines with the hemoglobin plus carbon dioxide, that is carbaminohemoglobin, that is around 23%. But maximum will undergo this reaction, that is carbon dioxide combines with water in the presence of carbonic anhydrase enzyme to form carbonic acid. So this is a very unstable compound. So it dissociates into HCO3 minus and H plus. Okay. So this H plus will be buffered by our hemoglobin. So one of the important function of our hemoglobin is buffering. It has a capacity of buffering. So immediately it combines with the H plus and buffer and maintains the pH. Then this carbon dioxide diffuses to the plasma for an exchange of chlorine. Chlorine enters into the RBC. For each bicarbonate which diffuses into the plasma, one molecule of chlorine enters into the RBC. So this is called as chloride shift, which occurs during carbon dioxide transport. Okay, so chlorine moves from the plasma to the RBC. This is called as chloride shift. Okay, so that's what I told you. Bicarbonate leaves the RBC in exchange of chlorine. That is called as chloride shift. and is transported in the lung in the plasma. H plus is buffered by deoxyhemoglobin, because deoxyhemoglobin is a better buffer and H plus than oxyhemoglobin. Okay, so in the lungs, all of the previous reaction occurs in reverse. So these all things occur in the tissue levels, and in the lungs all of the previous reaction occurs in reverse. That is, bicarbonate enters the RBC in exchange of chlorine, and bicarbonate combines with H plus to form H2CO3, which decomposes into carbon dioxide and water. Thus, carbon dioxide originally generated in the tissue is expired in the lungs. Okay, what I showed you in the tissue level, reverse of that occurs in the lung level. Okay, so this is the carbon dioxide dissociation curve. So this is the normal range. So PCO2 ranges from 40 to 45 or up 45. That's all. Okay, so this is the normal range. Okay, so normal blood PCO2 ranges between the limit of 40 mm of Hg in arterial blood and 45 mm of Hg in the venous blood, which is a very narrow range. That is, the concentration raises to about 52 volume percent as the blood passes through the tissues and falls to about 48 volumes percentage as it passes through the lungs. Okay, so concentration raises to about 52, is around 52 volume as the blood passes through the tissues. Okay, so in tissue around 45 mm of Hg, then the carbon dioxide in the blood is around 45. Okay, around 52. Okay, whereas in the blood passes through the tissue and falls to about 48 volume percent as it passes through the lungs. Okay, so in the lung it is 40 mm of Hg, then carbon dioxide in the blood will be around 40 mm of Hg. Okay, this is the carbon dioxide dissociation curve, but never in the exam carbon dioxide dissociation will be asked, but maximally it is asked oxygen dissociation curve. But you read and remember, you may get for your exam, I don't know. Okay, so now what is this Haldane's effect? The combination of oxygen with hemoglobin in the lung, okay, causes hemoglobin to become a stronger acid. So combination of oxygen with hemoglobin in the lung causes the hemoglobin to become a stronger acid. This displaces carbon dioxide. That is, the binding of oxygen to the hemoglobin causes displacement of carbon dioxide from the blood into the alveoli. This is called as Haldane's effect. Okay, whenever oxygen combines with the hemoglobin in the lungs, this causes displacement of carbon dioxide from the blood into the alveoli. Okay, this is called as Haldane's effect. Okay, now coming on to the effect of rate of tissue metabolism and tissue blood flow on interstitial PCO2. Okay, so this is the normal metabolism. Okay, so if there is a decreased metabolism, that is a 1/4 normal metabolism, and this is the 10th times the normal metabolism. Okay, so blood flow percentage of normal blood flow, and this is the interstitial fluid PCO2. Okay, here what happens? The decrease in blood flow from normal point A to 1/4 normal point B. So decrease in blood flow from here point A to point B increases peripheral tissue PCO2. So blood flow decreases from the normal level, that is point A to point B, then interstitial fluid PCO2 will increase. So A, it is around 40, it is around above 40, isn't it? But here it is above 60. normal value of 45 to an elevated level of 60 mm of Hg. Increasing the blood flow. So here it is 100. So it is normal blood flow. If it is increased to 6 times, 100 to 600 in a point C, okay, decreases the interstitial PCO2 from normal value of 45 to 41. That's what is telling. If blood flow increases to 6 times, then the PCO2 is 45 here, it will reduce to 41 at point C. Okay, so PCO2 level will decrease. That means normal blood flow, interstitial fluid PCO2 will be normal. If the blood flow decreases, interstitial PCO2 will increase. If blood flow increases, interstitial PCO2 level will decrease. That's the meaning of this graph, and this is the effect of metabolism. It occurs in our PCO2 level in interstitial fluid. Okay, so that is about the carbon dioxide transport and oxygen transport in our blood. So all of you go through once again, and make the notes. Oxygen transport is very important compared to the carbon dioxide, but chloride shift has been asked many times. Okay, so read about those things, make your own notes. If you have any doubt, anytime you can ask me. Okay, thank you.
Thank you, ma'am.
So any doubts?
Okay, ma'am.
You understood na? Oxygen transport and carbon dioxide transport. Hmm? Understood? So all of you go through whatever textbook you are having, go through once, okay? Then definitely if you read, then only you will get the doubts, isn't it? If you don't read, you don't get any doubts. So my honest request for you people is today only you go back and revise this or read this whatever textbook you are having and try to make your own notes. If you got any doubt, note down and you can ask me. Okay, thank you. Anything you want to say? Okay, I'll give them.